we have 2 more speakers. Thank you.
So I'll begin already. I'm going to get into something very interesting. I hope that I'll convince you that's super interesting about bird malaria and what we can learn from studying avian malaria, which is a very related parasite. So there are some thoughts that are becoming more clear that bird malaria is actually basal phylogenetically an ancestor to falciparum. So if you look at a phylogenetic tree of some of these parasites, you'll see that falciparum and the ape parasites such as Leverania and Reichenauii, for example, they seem closer to the bird malarias than does vivax, for example. And so there's some interesting thoughts about that. First, in terms of ancestral relationships, and then also what can we do with bird malaria? Of course, we can do a lot of things that you can't do with human malaria because birds are not subject to these socioeconomic things that we are. So it's a more natural system. And I just wanted to remind you about some interesting things that came out recently about malaria in humans, and that if a human apparently has a high infection of malaria, they're more attracted to mosquitoes. Now that's super interesting, right? And so now a couple studies came out from France that basically showed it's true. If you have birds infected with malaria, these birds actually attract more mosquitoes. And so now what these scientists are trying to do is figure out what is the compound that is attracting those mosquitoes. It's not CO2, it's probably something on their skin, some kind of smell, some olfactory cue. So, oh shoot, this didn't seem to work.
These are really dark.
I don't know, what happened? Maybe it has to go into
Why don't we please,
what'd you say?
we'll have to restart.
Oh, maybe.
It's the projector.
It's probably the projector, isn't it? So what we're doing at San Francisco State University, I'll tell you a little bit about myself too. So I actually did my PhD at UCSF, where Vita is doing her PhD in cell biology. But I got very interested in ecology and evolution of malaria as a postdoc, and for a couple of reasons. First of all, my sister used to do malaria work in Papua New Guinea. And then my aunt actually died from falciparum malaria, which it was a real tragedy. It was in 2000, and she was in India and went to a village, and they thought it was a fever. And she was treated by a Tibetan holistic doctor, and she died of malaria. You know, this is unheard of in New Delhi, which is not a big malaria capital. And so these things led me to think about how I can combine ecological questions with disease questions. And so I started beginning some work on the ecology of disease. And so this is how I got into my avian malaria research. And so I started working in Africa, and now I've also gone into other areas. I'm actually going to Peru on Friday to work in the Amazon, studying some malaria in birds in the Amazon and deforestation. So my big question is, what happens with these big rapid changes that are happening on the planet, such as deforestation or climate change? How does that affect malaria? And this is, again, a nice system because we're looking at birds. I don't need to even show slides. I can talk forever. So what we're doing is, I will show you some interesting slides, though. So we're looking at habitat changes, climate changes in Africa first. So one of the things that we began with was to look at how does habitat affect host specificity. So what's another thing that's really nice, so remember Knowlesy, that's a parasite that's jumping from macaques to humans in Southeast Asia. This is a new, relatively minor parasite, but it's jumping from these macaques to these humans. Now, why is that happening? What allows that? What facilitates that? So it turns out in birds that we have a lot of generalist parasites and specialist parasites. So we have this one bird called an olive sunbird in Africa, and we've collected hundreds and hundreds and hundreds of that bird, and it has one malaria parasite, and we never find it in another bird at all. Okay, it's just like falciparum is really specific to humans. Okay, then we find other generalist parasites that jump into lots of different birds. Okay, and so they are the generalists. We can count on them in lots of different birds. So we have these specialist parasites, we have these generalist parasites. And so, oh, there we go. And so now the question is, how does habitat affect that? These are all the people that I'm working with. We have people at San Francisco State University, and of course, a lot of ecological scientists and evolutionary biologists and malaria scientists throughout the world. So habitats and climate are changing. I talked about that. Birds are a great malaria model system. They have a lot of the same diseases that humans do, and we have a lot of information about birds. This is Hawaii. You've been to Hawaii, so you've seen the birds in Hawaii, but those birds are not from Hawaii. The ones that are actually in Hawaii are the ones on the tops of the mountains, because there's no mosquitoes there. So what happened was mosquitoes came to Hawaii about 1826, and with that came malaria, and it wiped out the birds in Hawaii. It was a tragic story. This little bird here, the ho'ouli, went extinct in 2004. So these are really affecting the birds. And again, with climate change, the mosquitoes are thought to be moving up the mountains and will kill those last populations of these Hawaiian honeycreepers, which diversified even more than the Galapagos finches. They're a tremendous, interesting malaria story, and poor things, they all die. This is an EEV, and this is an apapani. There's a mosquito on it. So now malaria in birds is different in some ways. And there's some cases where it's killing the birds, and some cases where it's not killing the birds. In most cases, it's not killing the birds. So we have Plasmodium, Hemoprotis, and Leucocytozoa. These are the Hemosporidium parasites, which you never study in humans, because we don't get these two. We only get Plasmodium. But these are very, very huge populations of parasites that affect birds. I won't talk about any of these other guys that are different types of things. But look at this tree here. Okay, if you look at this little red area, I don't know if you can turn this light off, but if you look at this red area, these are the human-invading parasites. There's only 5 human ones, right? Maybe there's 6 now. Ovale's been divided, right, into 2. So there's just a few. But look at all these blue ones. These are all avian malarias, okay? And it's quite clear that we have these are ancestral. At least in some of the recent phylogenies, we know that Plasmodium gallinaceans, the chicken parasite, is basal in the phylogeny to some of these human ones. And so then we also have Parahemoproteus and Hemoproteus, and they're spread by different vectors. So we have Hemoproteus fed by these biting midges, Plasmodium, of course, by mosquitoes, and Leucocytozoan by black flies. So you never hear about these parasites if you're studying humans, because they don't really affect us. But these are really great model systems for studying malaria. We're working on a lot of projects. We've described a lot of new species. We're looking at deforestation and prevalence, predicting prevalence in Africa, host specificity in habitat. I'll talk a little bit about that. Host-parasite coevolution. What is this jumping of parasites? How does that happen? Land use changes in Costa Rica, climate change, and then this molecular basis for host specificity. This is something that I actually started working with Vito a little bit about a while ago in the Derisi lab. We were doing some of that work looking at transcriptomes. So we've described a lot of new parasites. These are, remember that red blood cells in birds and frogs and reptiles have nuclei. This has been our biggest nemesis, actually, because it's been very hard to get pure DNA from these things. What we're doing right now, if you know this life cycle, is to purify microgametes. And so we can purify microgametes under exflagellation. We can use that for getting sequence data. So we've described a lot of new parasites from Africa. And so here you see these parasites inside the red blood cells. So habitat and host specificity. This is interesting. So we've collected some samples from lowland rainforests, from higher mountaintops, and from this region that's kind of like California and South Africa called the Fynbos. And all right, so we've developed these host specificity indexes. These are mathematical constructions based on phylogeny and relationships. And we can figure out these host specificity index. The lower the number for host specificity is the more specific they are. So these indices of host specificity, these are the specific ones, these are the generalist ones. So it turns out that if you're in a very stable climate, such as in the lowland tropical African rainforest, where you don't see much changes in temperature, you have these very specific parasites. Okay, so it makes sense. The parasite can be able to find its host, no problem. It's very specific, very stable and very predictable. Whereas if you're in the highlands, you're going to have more generalists. And so the generalists have to be able to find their hosts. And if you can't have a predictable environment, you might not find your host. And so that's why they've developed into being generalists. Same thing with host range. So if we have a huge host range, like a sunbird in Africa, an olive sunbird, which we study, you'll have more specialists, right? These specialists will be able to find that bird in all these habitats. But when you're looking at, again, these mountaintops, like these highlands, for example, you're going to have to find generalist parasites because the generalists might not be able to find this bird very much. And so that's why it's kind of tipped the edge in evolution towards that generalist mode of evolution. And what's interesting is when we do these phylogenetic reconstruction analyses, we find that generalists become specialists over evolutionary time. Okay? So what that means is that when you look at these parasites over many, many thousands of years, such as the human ones, for example, they're becoming more specialized. And what does that mean for virulence? That's a big question. We don't know. That's one of the things I'm really interested in, is how does this affect the virulence? Specialists or generalists? Which is more virulent? You can argue either way in some cases. I mean, the one that got into Hawaii was a generalist and killed all birds. But you can also argue that a specialist might possibly, like falciparum, that's a specialist, right? It's only in humans. That might be a dangerous parasite. Look at this. This is the sunbird Plasmodium co-phylogeny. What a mess. Okay? What this means is that the parasites are jumping from host to host in the sunbird family a lot. There's a lot of this jumping between the parasite and the host. So that means there's host switching quite a lot over evolutionary time. These are all the different plasmodiums that we've seen, and these are the sunbirds that we've seen. This is one family of birds. So it's kind of like thinking about apes or something. These are really related birds. If you see some other species of birds, if you added more genera and more families of birds, you'd see some more host specificity there. But what we're seeing here is this real tanglegram of these parasites linking to their lineages. And so what we're saying is co-speciation alone does not play a significant role in the diversification of Plasmodium. Host biogeography or habitat contributes to the structure of avian malaria parasite communities. So what that means is that the relationship with the host is not what's dictating the speciation, right? It's basically other things are also involved, such as habitat, biogeography, and these things play a big role in the evolution of malaria. So a couple things. This is Costa Rica. We looked at some agricultural land there, coffee plantations. You've all bought bird-safe coffee, right? And so buy that. That's good stuff. We looked at forest cover, and so we have areas where there's high forest cover and low forest cover, and looking at these malaria parasites. And so I will go through this quickly, but basically, parasite lineages do not differ by habitat. We see hemoproteas, we see malaria parasites. If you're looking at the different types of habitats, you find all the lineages around them. All those ones are there. But what's interesting here is that we have this one bird, it's called the orange-billed nightingale thrush, and we studied that bird in depth, and when we do some modeling here, we found that when you add more forest elements, meaning trees, to this agricultural land, which is this purple line here, it reduces malaria in those birds. So it means that when you have more forest for that particular species of bird and that particular parasite, you have less malaria. It mitigates the malaria. Same with the rainforest, except if you add more trees, it's a little bit less of a slope here. But this is kind of radical, that it means that this forest elements, this forest habitat is really influential for what type of malaria is there and how much malaria prevalence there is. And so again, this has implications for humans, of course. Cutting down trees. Obviously, when I'm in the middle of the jungle in Africa, I'll never even think about getting human malaria. There's no humans there. And there's no mosquitoes that are flying from the villages 30 kilometers into the middle of the forest. And also, we're not getting gorilla malaria very much either. So what this means is cutting down this rainforest, changing these habitats is really going to affect malaria in birds and humans and rats and lizards and everything. Again, this is that land sparing versus land sharing. Land sharing means that you have the coffee plantations with plants on them with trees. Land sparing means you just cut down the plantation and you have the plantation next to the forest reserve And so in all of our models, you see that land sharing is a good idea for mitigating malaria. Okay? So you can see the relevance with humans here, right? I think it makes sense. One more thing, Alaska. Climate change. I was talking about climate change in the elevational gradient. This is climate change in the latitudinal gradient. So this is not Hawaii, but Alaska. So we're thinking about how will birds be infected at the higher latitudes with climate change. Of course, we're thinking that malaria mosquitoes are going to go farther and farther north. We have malaria in birds in Alaska. So here's where we collected birds, all these different regions. This is our most northerly region. We collected a lot of mosquitoes, but you know what? We never found a malaria-infected mosquito. Okay? There's just too many mosquitoes out there. Of the birds, we have resident species and we have migrants. And these birds, these three types of birds here, chickadees and red bulls, they are not moving. They stay the whole winter in Alaska, so they don't migrate. So we can use these birds as a good indicator of where malaria is being spread. So we now detect malaria below Coldfoot. So in Fairbanks we saw some, but never up here. We've gone through several years, several seasons. Up here we've never found malaria in birds, in the resident birds. Of course, there's some migrants that will have it. This is our future climate scenario with these IPCC numbers. And so by 2080, we expect that malaria will actually reach Coldfoot and even go a little bit north, but it will never get to this white area above. It won't get up there, even by 2080. So this is a prediction for where malaria will be in the future. We have also some co-infection data. We think that leukocytosome, that one parasite, trypanosomes, we have a lot of co-infected birds. This is something else that's kind of interesting that I think malaria human scientists should study more is these interactions among different pathogens. What is that going to do? And so here we're doing a little bit of that, trying to predict what these interactions of pathogens are doing for malaria and how the main indicator, again, is temperature. So temperatures are affecting these co-infections. Here's our maps for how we think that these co-infections will be affected. And these are our hotspots for these co-infections. So it's not only latitudinal or temperature driven, there's other potential drivers such as vector abundance. We're still studying this. Finally, a little bit before Vita talks about this transcriptome and the molecular basis for host specificity. So we know, again, that these parasites can jump hosts. Now, what's the reason they can do that? What molecules facilitate that? That's something that is very nice to study in these birds because we have, again, the generalists and the specialists. And so we can divide those groups up. But first, we just don't have very much data on these types of birds and their genes. So this is the only one that had a kind of rudimentary genome. This is a chicken parasite, Plasmodium gallinaceum. And so it's a well-studied model system. And again, people are at the Wellcome Institute trying to get a better genome of this. But we said, well, let's get the transcriptome. And so we started doing some of this work. Again, with Vito was helping us with this, and we finally got this to work. We've identified one gene. So people think that there's this class of genes called the erythrocyte binding-like genes. These are the proteins that are important for getting malaria parasite hematozoa into a red blood cell. And so we think that these genes are important for conferring the host specificity as well. And so we've identified we've only identified one of these genes in gallinaceum, and that is this MABEL protein. And it has very conserved gene structure to the falciparum one and some of the other malarias as well. And so it also has synteny, and we can look at these things. What we want to do now is look at gene expression between the specialists and the generalists. So now we have this gallinaceum transcriptome. Again, we've only seen this one EBL protein, which is a little bit surprising, but not completely surprising because falciparum has multiple pathways of erythrocyte invasion. There's EBA 175, EBA 181, EBA 140, there's a bunch of these things. But here, it looks like we have one. And you know, Duffy is the only one that's in 5X. So basically Duffy in 5X is the major one as well. So it's possible that there's only one. And so we're looking at this. We've got this transcriptome of the chicken one. We have some really good infections. We are working at Davis with Lisa Tell on this. So we got this gallinaceum transcriptome. We have a 81% showed homologs to be very generalist in its feeding patterns. And it seems to bite all the different birds. And so somehow it's not a filter. The mosquito itself is not the one that's doing this filtering. So I believe it is probably something on the parasite itself that's leading to this host specificity. So we're looking at homologs of falciparum genes, getting rid of the chicken transcripts a little bit more, because again, there's that contamination with chicken transcripts. We're looking at alternative splicing, genes and proteins involved in erythrocyte invasion. So that was my quick story. We have a lot of things to do. Vector ecology is a big black box here. We just don't know what's going on with vectors and how that is affecting these systems. For example, in Alaska, we'd like to know how is the vector involved in this. Is there just a missing vector in the far north, a particular species of, it's not only Anopheles that's spreading these things, but a lot of different culex and Aedes and Culecita, these spread the malaria birds. Transmission, we're looking at some California solvers, linking up the vectors in the Fresno area with Anton Cornell at Davis. We're looking at linking up the vectors with the bird parasites and figure out which ones which one's being transmitted by what. And then again, the last stuff on the evolution of erythrocyte binding. That's it. Thanks very much. I would be glad to take some questions, because this work is really interesting, and I think, yeah. Okay.
Just one comment and one question. We find that the changing ecology is affecting malaria transmission globally. So development has played a huge role in the reduction of human malaria in the last 20 years. In cities, we find urbanization, yeah, pollution. Essentially, it's pollution, but also in Southeast Asia, the biggest vector is a vector called Anopheles dirus that lives in the forest fringes. And of course, if the forest fringes no longer exist, then the vector doesn't exist. And so in Cambodia and Vietnam, the huge destruction of the rainforest has in fact been beneficial for malaria control. My question is really, you were showing us the stretch of malaria going north. Is that based on the cycle, the vector cycle being inhibited? We know with human malaria.
I'll just tell you my experience in cold fit Alaska. It is so variable. Like 1 minute is hot, the next minute is cold. Okay, so it just doesn't seem to be a warm enough temperature for the time that would require for the parasite to survive in a mosquito and develop in the mosquito. That's my prediction. Because boy, it was in the summer and suddenly it's like freezing. I'm in my tent, I need 2 sleeping bags. And then the next day it's like, 27 degrees Celsius. So and we know that malaria and these mosquitoes doesn't really survive below 13 degrees Celsius, or maybe 13, 14. And so if it's not at that level for a week or so, you won't have the proper development, right? And so I think that's what's going on in the far north. You just have this variable up and down, up and down. So I don't know if that answers your question. Yeah. But in Fairbanks, for example, you have some hot days for a long period of time.
For the transcriptome stuff you just talked about, obviously you went through it real fast, but I'm wondering why you don't just start with looking at sequences, compare sequences of EVL across.
We didn't find any other ones. So what we did first was look for EVLs in the genome of the chicken parasite, but that genome is very incomplete. And so first we were all, well, should we do the transcriptome or the genome? And so we decided to do the transcriptome because we got a very high parasitemia, and it's just a little bit easier, and then also we're interested in some of these splicing things at the same time. But still, it was interesting, even after looking at the genome plus the transcriptome, it seems like that MABEL protein is the pivotal protein here, the critical one for erythrocyte invasion. Because we know from human experiments that with falciparum that you can block invasion with peptides against MABEL, for example. And I think antibodies will block as well. But again, of course, falciparum is very complicated with several different invasion strategies.
I'm just thinking, if you have DNA from a lot of species, an easy project for a summer student would be to just sequence homologs in 25 different.
Exactly. So our problem is that these parasites are at chronic levels with very low parasitemia, so PCR just doesn't work very well from these guys because you have a very hard time getting enough amplification because again, there's thousands and thousands and thousands of DNA copies from the bird and just 1 or 2 copies of DNA from the parasite. This has been our nemesis, as I told you before. It's just we don't have enough parasitemia in most of these birds to get a good amount of DNA.
Can you inoculate them into culture?
He's like, okay.
Well, we know we can do, we can inoculate, of course, Plasmodium gallinaceum.
Yeah.
But we haven't tried with the other birds. And we're thinking, well, that's what Jenny Carlson's doing at Davis. She's one of our students. And so she's trying to get at least some infections to be transmitted to canaries and then bring it to Hawaii. So that's something we're really interested in doing. But I think you recognize this very interesting question about the specialists and generalists that we can really answer with this type of system, which would be very exciting.
Sure. What was the basis for the land sharing? Your hypothesis
Right.
about why that would reduce avian malaria burden?
So basically, based on our evidence in the field, when you have more forest trees in these coffee plantations, you have less malaria in that particular room. And so the land sharing means if you have more trees on these areas of agriculture, we predict there's going to be less malaria.
So just because you're recruiting more birds to the area?
Well, that's a good point, and maybe it has to do with this dilution hypothesis. So if you have more birds, it might dilute out the malaria from some of those birds. And so that's called the dilution hypothesis, where you just have more species of birds, and so that would, or more species of vectors, for example, which would kind of, and the parasite would get in the wrong host more often, that would dilute out the malaria. That's a good hypothesis for that. Thank you very much. I think it's Vita's turn.